गुड मॉर्निंग स्टूडेंट्स। आज डिस्कस करेंगे सेकेंड पोस्ट फर्टिलाइजेशन इवेंट्स दैट इज़ एम्ब्रियो। एम्ब्रियो का स्ट्रक्चर, टाइप्स, डेवलपमेंट सब यहाँ पे डिस्कस करेंगे। सो फर्स्ट एम्ब्रियोजेनी। एम्ब्रियोजेनी इज द सम टोटल ऑफ चेंजेस दैट occur during the development of a matured embryo from a zygote, and these are the various diagrams for development of matured embryo from a zygote. This is zygote. Zygote divided to form two cells, suspension cell and embryo cell, and the suspension cell to form 6 to 10 cells. And this is the formation of suspension, and this is the first cell of suspension. It acts as ostomium. This is last cell. Last cell is called hypophysis. Then embryo cell also divides to form 8 cells, outer 4 cells and inner 4 cells. And these are globular embryo. It is globular embryo. This is odd-shaped embryo, and these are the matured. This is matured embryo, and this is the full structure of dicot embryo. अब डिस्कस करेगा embryogeny in dicots. In a typical dicot, the zygote elongates and then divides by transversely to form two unequal cells. The larger basal cell is called suspension cell, and smaller terminal cell is called embryo cell. Again, the suspension cell divides transversely to form 6 to 10 cells. The suspension helps in pushing embryo in the endosperm. The first cell of suspension towards the micropellar end acts as ostomium. The last cell of suspension towards embryo is known as hypophysis. The later hypophysis give rise to radicle and root cap. Then now the embryo cell undergoes two vertical divisions and one transverse divisions to form 8 cells, and 8 cells arranged in 2 tiers. This is called octant stage. It is also called octant embryo. Then outer 4 cells are called epibasal cells, and inner 4 cells are called hypobasal cells. Again, later the epibasal cells eventually form the two cotyledons and plumule. So cotyledons and plumule produced by outer epibasal cells. Then hypobasal cells produce hypocotyl except its tip. Then the octant embryo divides periclinally to produce an outer layer of protoderm or dermatogen. Protoderm later give rise to epidermis. Then inner cells differentiated into procambium. Procambium and ground meristem. Protoderm forms epidermis. Procambium gives rise to steel or vascular strands, and ground meristem produces cortex and pith. Initially, the embryo is globular and undifferentiated, and this type of embryo is called proembryo. Then proembryo is transferred into odd-shaped. Odd-shaped embryo becomes matured, and matured embryo has radical plumule and two cotyledons. And this is the structure of dicot embryo. So a typical dicot embryo has an embryonal axis and two cotyledons. The upper part of embryonal axis is called plumule, and the lower part is called radicle. The point where plumule attached with cotyledons is called epicotyle. The point where attached to radicle, the point where radicle attached with cotyledons is called hypocotyl. An embryonal axis is also called tigellum. And this is the embryogeny in monocots, and these are the respective diagrams, and this is the structure of monocot embryo. This is scutellum. The scutellum is also called single cotyledon and coleoptile. This is shoot apex. This is epiblast. This is radicle, root cap. This is root cap, and this is coleorhiza. This is the structure of monocot embryo. And in this process, the zygote elongates and then divides transversely to form basal and terminal cells. The basal cell produces a larger swollen suspension cell, and suspension cell it may function as ostomium. The terminal cell divides transverse division to form two cells, top cell and middle cell. Then the top cell forms plumule and single cotyledon, and middle cell forms hypocotyl and radicle. And this is the structure of monocot embryo. The embryo of monocots have only one cotyledon and embryonal axis. And in monocot embryo, cotyledon is also called scutellum. It is a large shield shaped. Then upper part of embryonal axis is called plumule and lower part is called radicle. Both plumule and radicle covered by coleoptile and coleorhiza respectively. So this is the structure of monocot embryo.